is all also over time a useful criterion to work with. Then panel D and E, they show the reconstruction of the trajectories followed by the monkey's hand, reaching eight possible targets, like clockwisely. They are labeled there, and you see the different trajectories in color, and the green, blue, red, and so on. So, and then you see the correlation coefficient, so that's the Pearson's correlation between the actual trajectory performed by the monkey and the trajectory predicted from in this case, local field potential recordings. And you see a 74% correlation there. If you look at spiking activity, you see a bit higher. It goes 93, it's higher. But if you compare in plot F, the spread of the accuracies of position and speed, then you see that there is not that much significant difference between the different modalities, and in terms of speed, local field potentials are actually doing better than the other signal modalities. So this shows, and to summarize, this shows that local field potentials indeed can be used for decoding information recorded from the brain, in this case, movement of hand. Then you can start developing the idea. What we did with an experiment here in the lab with a colleague of mine is to actually look at plasticity effects. Remember last class at the end we indicated the effect of plasticity. When you're learning a task, you're getting better at it, and this is revealed by the tuning curve, so the firing behavior of your neuron. But this is also the case with local field potentials. And the experiment was as follows. So a monkey was being trained to make the distinction between target and non-target stimuli, target being a stimulus for which he will get a reward, a juice reward, versus a non-target stimulus, which in this case he doesn't get any reward. You see that after some days, we will show it shortly, after some days you get indeed a clear improvement based on local field potentials. And this is a known phenomenon. It has also been observed in other brain areas in terms of local field potentials. And this way we know that this will work. Okay, so let's now go a bit deeper in just what I've been saying. So this is a so-called visual brain-computer interface, so based on local field potentials. Okay, so I'm now on slide 38. So the Utah array has been implanted. You already know the Utah array, so it's developed by, as the word says, University of, well, if it's the term suggests, it's University of Utah. They give it their own name, the name of their state. And yeah, it's a 4 by 4 millimeter array with 100 electrodes, etched out of silicon, spaced by 0.4 millimeter and 80 micron in diameter. And the tips are metallized with platinum. So it's only the tips, as I said before, that will record activity. So these Utah arrays can be used for recording spikes, of course. That's what they in first case are intended for, but they can also record local field potentials. Remember that what you're recording is a sort of raw signal that can be split after filtering in a local field potential. So the lower frequencies, and the higher frequencies, they correspond to the action potentials or spikes. So two monkeys have been implanted. And in page 39, you see an image there. So the opening in the skull of the monkey, and you see this black square. That's the implant. And there are some landmarks there, in terms anatomical landmarks, and these are sulci that that you can use in order to figure out that you're recording in the area you want to record. In this case, it's visual area number 4. And if we move to the next slide, number 40, that shows the stimuli. So you have 2 types of stimuli. So it's a grading, so it's a sinusoidal ondulation. It looks a bit like a waffle ondulation, and that has a particular orientation. So in this case, 11.2 degrees, and on the right side you have the opposite case at 22 degrees, and there were 2 monkeys implemented. So the other monkey had a different orientation combination. So everything looks a bit noisy. This is done intentionally, so there is noise added to the two stimuli, and to make the task a bit harder so that the monkey would be paying attention. And this kind of noise is called sinusoidal white noise. I mean, the term itself is not so important, but it sort of occludes the task and make it more harder for the monkey to distinguish the 2 types of orientations that have been that are presented. So in slide 41, you see then the sequence of events. 500 millisecond there is a stimulus shown, and then 500 millisecond the next stimulus, and then you see that there is also simply noise there, and the monkey has to look at the red dot in the middle of the screen. There is noise there, and there is stimuli that are unrewarded, and if there is a rewarded stimulus 400 millisecond after stimulus onset, the monkey will get a reward. There is no other task than explicit task, right? Just the monkey needs to pay attention to the red dot, and that's it. And of course we do recordings then, and from those recordings we can try to predict if this was a stimulus for which reward was given or not. So the monkey actually a reward expectation, can we pick up that expectation from our recordings? As I said, there were 2 monkeys. There is conditioning happening, as I just indicated, 37 days for monkey 1, 55 days for monkey 2. Suddenly, the pairing was reversed. So the stimulus that was unrewarded before became the rewarded one, and vice versa. Of course, in the beginning, the monkey is completely missing out on that, but after some training, further training, the monkey picks that up, that the switch happened, and the whole model reverses. And this is clearly observable with also in the decoding case. So this then continues this switch after more days, 20 days, monkey 1, 52 days for monkey 2. Okay, so now the task for ourselves, not for the monkey, the task for ourselves is to build a brain-computer interface of which the classifier can predict the type of stimulus, if this is stimulus for which reward will follow or not. And to be fair, we only use the first 300 milliseconds after stimulus onset, because remember, at 400 milliseconds the reward comes, otherwise you would be modeling probably something else, the juice intake of the monkey, I mean, over the muscular stuff. So this is not a fair task. Then it would, what we want to do is to consider it visually. So, of course, this is a nice paradigm itself, and you can imagine that you could use this online to monitor the training performance of the monkey in real time, and adjust your juice reward or your training duration and stuff like that. So, but we didn't go that far. Okay, next slide, slide 44, you see the local field potential waves that correspond to the rewarded and unrewarded case. You see the blue curves are single trials, so you see a lot of wiggles there, and the green one is another trial. These are single trials for electrode number 80, for example, from day 36. There is not much to be discerned between a rewarded and unrewarded case. So this is actually what we work on, single trial case. So you see the difficulty of the task. Of course, if you average over 100 or so trials, these are the red curves, then you see a difference in how the amplitude evolves over time, but this is not what we do. So we actually consider decoding individual trials. You see how hard that is. And on the bottom panel, you see